Friends, further we will study the vegetative cell in male gametophyte. After pollen mitosis, the vegetative cell continue to grow. The cell organelle increase in number as well as in size. The vacuole gradually disappears and the RNA and protein content of the cell increases dramatically. The nuclear envelope becomes highly convoluted. Normally, the vegetative nucleus remain arrested in the G1 phase of the cell cycle and does not synthesize DNA. At maturity, the nucleus often lack a nucleolus. In Hemanthus, the large nucleolus initially present is replaced by a small nucleolus. In certain orchid, also the vegetative nucleus possesses a nucleolus. According to Sanger and Jensen 1971, in the development of vegetative cell in Hemanthus, four distinct stages can be identified. Immature stage, during which the nucleus begins to lose its spherical shape. Mature stage, which is characterized by an increase in the number of dictyosome and change in the staining density of lipid body from dark to light. Starch accumulate in the plastids. Stage shortly before anther-dehiscence, during which the nucleus is highly lobed, lipid bodies are absent, and the plastids no more contain starch. Pollen tube stage. Next is the generative cell. Immediately after becoming detached from the pollen wall, the generative cell is spherical. It undergoes a lot of change in shape during the pollen development. Usually the cell elongates, sometimes to such an extent that in mature pollen grain it appears vermiform. The elongated shape of the generative cell probably facilitates its movement into the pollen tube. The elongation of the generative cell has been attributed to the microtubules, which are oriented longitudinally parallel to the long axis of the cell. Sanger and Jackson 1971 have shown that if a pollen grain containing ellipsoidal generative cell is treated with a substance such as colchicine or isopropyl and phenyl carbamate, which selectively destroy the microtubule, the generative cell tend to regain its original spheroidal. The generative cell cytoplasm is highly reduced, but it contains usual cell organelles: mitochondria, ribosome, endoplasmic reticulum, microtubule, and dictyosome. While most of the species investigate, example beta, Hemanthus, Petunia, lack plastids in the generative cell, and there is evidence of being present in some other species in Oinothera hookeri, Medicocarpus sativa. When present, the plastids do not contain starch. The mitochondria in the generative cell are poorly developed, except in barley, where they show well-developed internal membrane system. Formation of sperm. Sperms are formed by the mitotic division in the generative cell. This may take place after the release of the pollen from the anther or while the pollen are still confined to the anther. Thus, in the former condition, the pollen are shed at the 2-cell stage, and in the later at the 3-cell stage. In those plants where pollen grain are shed at the 2-cell stage, the generative cell may divide inside the pollen grain after it has alighted on the stigma. As in case of Holoptilia intrigifolia, or in the pollen tube before it reaches the embryo sac, or really after the pollen tube has reached the embryo sac in Euphorbia teresiana. Generally, DNA synthesis in the generative nucleus starts immediately after the pollen mitosis. This is followed by a more or less prolonged mitosis. During this period, the generative cell undergoes elongation to acquire its characteristic vermiform appearance. In the species with binucleated grains, the division of the generative cell is postponed until the pollen germination, and it passes through a stage of temporary dormancy with its nucleus in the mitotic phase or really in the metaphase. In case of impatient balsamia, a kind of suspended mitosis. In trinucleated grain, on the other hand, the generative cell divide within the pollen grain by essentially a normal mitosis. The three cell pollen grain are released during the M phase, having two nuclei, sperm nuclei. In the two cell pollen grain, the generative nucleus remain undivided and contain double the amount of DNA of the sperm nucleus. Hence, the nucleus divide in the pollen tube. Thank you.